Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to today's webinar titled Innovative Rabbit Antibodies and Engineering for Therapeutics and Diagnostics, featuring Dr. Toshi Maruyama, Chief Scientific Officer, and Dr. Kevin Ensminger, Director of Antibody Discovery and Engineering at Abwiz Bio. Today, they will present innovative methods for developing rabbit monoclonal antibodies for therapeutic and diagnostic applications, showcasing cutting-edge phage display and antibody engineering techniques. I wanted to take this moment to thank our sponsor for this event, Abwiz Bio, for making it all possible. All right, thank you so much for being here, guys. And Toshi, you can take it away whenever you're ready.
Okay, thank you very much for the introduction, and I'd like to thank everyone for attending this webinar today. So I hope this one will be an informative one and educational one for you. And in this webinar, you can learn why rabbit antibodies are good for therapeutics and diagnostics, and that phage display is the best method for the isolation of antibodies when it is used properly, and how a unique antibody engineering method, stage-enhanced maturation, STEM technology works, and the durability assessment and optimization using this technology. So let me start with the topics that we're going to cover today. First, why rabbit antibodies and the importance of immunization and plasma cells, and of why this phage display, and a live reconstruction method WIZAMP, and a phage display system Abwiz, and a proprietary humanization designs, and the stage enhanced maturation STEM technology for antibody engineering, and the durability assessment for therapeutic development. So let me give you a brief company overview first. Our company was founded in 2012 based on a monoclonal antibody technology using phage display, and have over 30 years of experience in the development of numerous monoclonal antibodies from immunized human donors for infectious diseases and from immunized mice for cancer targets by phage display, including phage 52, which is the first human neutralizing antibody against the Ebola Zaire virus from the convalescent patient bone marrow samples, and the samalizumab, which is the humanized anti-CD200 mouse antibody that progressed to clinical trials. And our business activities are contract projects from the industry and a collaborative therapeutic antibody development with academia. Our core technologies are patented antibody gene cloning method with proprietary phage display system and antibody engineering technology. All operations are conducted in the United States, and animal immunization is performed at facilities in Denver and Pennsylvania. From live reconstruction to antibody discovery and IG production purification, all processes are carried out in-house without outsourcing to third parties. So first topic is why rabbit antibodies. So rabbits can respond to antigens that are challenging for mice or humans, producing antibodies with exceptional specificity and high affinity. Why? Because they go through more rounds of affinity maturation via somatic hypermutation and gene conversion in the germinal center. And also they have more diversity in VH-VL combinations. I'm going to explain this one in the next slide. And so that's why they can acquire picomolar affinity, which is superior to mouse and human antibodies in nanomolar. And also they have reduced self-tolerance. They can recognize poorly immunogenic and conserve the human antigens, even those with 100% sequence identity with rabbit proteins, and identify epitopes missed by mouse antibodies. So all these together are good for targeting human antigens and therapeutics. They're also good at detecting subtle epitope changes, such as post-translation modifications, PTMs like phosphorylation, methylation, sulfation, and glycosylations, and also single amino acid substitutions, cryptoproteinases like a new epitope made by the enzyme digestion, also idiotype of an antibody, small molecules, paralogs, isoforms, and subtypes. So this one shows the diverse VH and VL combinations in rabbit antibodies. This is from a publication. Rabbits has more longer and more diverse amino acid length distribution in both heavy and light chain CDR3s than mice. And these are all antibody sequences from mouse antibodies and rabbit antibodies. As you can see, the LCDR3 of mouse antibodies is restricted to 9 amino acids, and the pattern is poor, even though they're against different targets. So basically, the heavy chain CDR3 is the determinant of affinity and the specificity. So it's like the mouse. In mice, VH is the main drive to antigen, bind to the antigen, versus rabbits have more diverse lengths and patterns in both LCDR3 and HCDR3. Sometimes they have two cysteines in LCDR3, like in human DH region. So it's like rabbits are trying to grab the antibodies, antigens with two hands with more complex combinations. That's why they can acquire higher affinity specificity and more epitope recognition. So the next one is the importance of immunization and plasma cells. So as you repeat the immunization, affinity and specificity are enhanced through affinity maturation in the germinal center. They start with IgM producing naive B cells, and the class switch to IgG producing memory B cells, and eventually they become plasma cells that sit at the top of the antibody producing pyramid, generating the highest affinity and most specific antibodies. They are terminally differentiated B cells, and they have the highest affinity and also secrete massive amount of antibody, which we see in the serum after the immunization. And after the immunization, most plasma cells migrate to the bone marrow for long-term residence. That's why it is important to collect bone marrow and spleen cells to acquire as many plasma cells as possible. But do not express surface IgG, meaning that they cannot be sorted by surface IgG expression and by negative and positive selections with antigens, like in single B cell cloning. So we do phage display. So why? Because with immunized rabbits, we can select antibodies with higher affinity and specificity by various selection strategies, which is unlike clones from naive or synthetic libraries, often do not have the function or have low affinity and specificity that need further engineering. So we collect bone marrow and spleen cells, isolate RNA, and amplify antibody genes and make the library, and produce phage in E. coli cells. And each phage expresses FAB fragment, which is the binding portion of the antibody, fused to phage through gene 3 protein, and the phage contains DNA. So phenotype and genotype are always connected, so you can mix them together and allow binding to the target antigen. And wash away non-binders and also remove cross-reactive clones by competitive selection, which I'm going to explain in the next slide. And the bound phage is eluted, and then more phage is produced in E. coli. So this is the procedure called panning. Repeat 3 to 4 rounds to enrich with the specific binders. And now you can screen one by one as a cerebral fab in ELISA, flow cytometry, functional assays, and Western blot. And since phage contains DNA, you can amplify antibody genes by PCR and then send a fragment for sequence analysis. And next day, you get the sequence data and then match with the binding and functional data and then pick the best one for IgG expression. And you can use that PCR fragment, cut and paste into the IgG expression, IgG vectors for expression and purification. And there are many selection strategies available for phage display. That's not achievable by screening alone. For example, competitive selection is a really powerful way. When you see strong reactivity after the immunization, then you can remove all the cross-reactive clones, such as when you immunize with fusion proteins, such as like FC and GST, you can use those proteins. And also, if you're targeting idiotype of an antibody, you can use a human IgG and also small molecules, paralogs, isoforms, subtypes, anything you can mix with your phage in a soluble form and absorb and remove all the cross-reactive binders from your library. And select the ones that are specific to your target, which could be small molecules, peptides, recombinant proteins, or cells in any combinations. This is really powerful to remove all the cross-reactive binders from the library, and have a publication on this one. Another important method is the epitope masking. When you do the immunization, there are always dominant epitopes on the antigen that more antibodies are produced against, but they may not have the desired functions such as blocking or neutralization. But you can use those to mask the epitope on the antigens to block the phage clones that bind to the same epitope. Then you can force the selection of binders to other epitopes on the same antigen. This is really good to select binders to various epitopes, and you can create a whole new panel of antibodies from the same library. So I have a publication on this one too. But there are issues with traditional phage display. First of all, you have to amplify antibody genes to make the library. By conventional 2' PCR, there's always a biased amplification of antibody genes. First of all, there's a primer efficiency. Some sequences are amplified preferentially, while others are underrepresented or even lost. So that skews the library diversity. And also, there's a melting temperature differences between forward and reverse primers that can lead to non-specific amplifications and the primer dimer formation. And the typical library size is restricted to 10 to the 7 to 10 to the 9, which makes it difficult to fully capture the antibody repertoire for well-immunized animals. And there are more issues with the traditional phage display. First of all, E. coli doesn't like mammalian antibodies. E. coli cannot propagate for Fab fragments due to multiple interchain disulfide bonds. This leads to mutations and deletions in the antibody sequence by homologous recombination and replication stress. And furthermore, most of the rabbit antibodies use CKAP1 light chains that contain cysteines in framework 3 and constant region, forming stabilizing disulfide bonds. And also there are consecutive two cysteines in the CH1, further reinforcing antibody structure. But this creates a huge problem for the E. coli cells to propagate for Fabs, leading to cell deaths even before they can express them on phage, making rapid antibody display and selection very difficult. So after 5 days of panning, you don't get almost nothing. So we came up with our own library construction method, WizAmp, which is a patented method that adds artificial sequence to both cDNA ends that can be used as a primer in a single primer amplification. And the annealing temperature is really high, same as the extension temperature, 72 degrees, that allows really robust and uniform amplification of every gene and enables faithful and comprehensive coverage of the antibody repertoire from the immunized animals, achieving up to 5 times 10 to the 10th clone from a single rabbit. That far exceeds the diversity of screening by hybridomal single B-cell cloning. And this applies to all the other species. And we also have our own phage display system, Abwiz. It's an advanced phage display system with the engineered phage meat vector and a panning protocol to fully support the selection of rabbit Fabs with CKAP1 light chains that traditional systems struggled with. And it is compatible with all the other species, including VHHs of alpacas and llamas. They sometimes have cysteines in CDR2 and CDR3, which are believed to contribute to the stability and affinity, but the expression in E. coli could be challenging. So with the same samples that didn't get any binders, with this system, we get many binders, but not only that, there was a marked increase in the diversity of the antibodies we obtained. So we got this ARC1-2 phosphorylation specific antibody, and we use a modified native Fab instead of SCFV. So there are issues with SCFVs. First of all, you have to amplify VL and VH by traditional PCR, 2' PCR. You already have a biased amplification. By linking VL and VH by overlap PCR, you introduces more PCR bias and also potential PCR errors. That affects the antibody diversity. And also, this is a big problem. There's a free unpaired cysteine in the framework 3 of the light chain. That could cause aggregation, poor expression, and reduce stability. And there could be loss of binding when SCFV is converted to IgG. So overall, it affects the diversity and quality of the antibodies obtained. And we do panning and screening fully automated by 2 custom-made robots. This machine handles panning in a 96 cell plate format, 16 conditions or 16 libraries simultaneously. And the other machine handles screening and even DNA purification and antibody purification. And Kevin will explain how these robots operate and handle panning and screening in a short video presentation later. So we do screening by ELISA and a flow cytometry in a 96 cell format, and also affinity measurement by ARC-TET and Western blot using a fluorescence dissection system. So in summary, minimizing the loss of valuable clones at every stage is the key to obtain a higher success rate. So in other words, if you do any step bad, you will lose your important clones forever. So you have to start with a good immunization and proper evaluation of the serum and collect as many plasma cells as possible from bone marrow and spleen, and to capture the antibody repertoire faithfully into your library and efficiently express FAB on the surface, not by screening, by selection. With various selection strategies, you can obtain a larger pool of clones that you can select the lead candidates from. And this is timeline. It takes about 2 months for the immunization and the serum analysis, and 2 to 3 weeks for the library construction, depending on the purpose of the antibody discovery, and also 2 weeks for panning and screening, another 3 to 4 weeks for IG cloning and production. So in about 4 months, we can obtain initial lead candidates as recombinant IgGs. So I show you some example using this system for the discovery and the development of rabbit monoclonal antibodies for multi types of antigens and purposes. The first example is a therapeutic antibody development against IgE for allergic diseases. There's omalizumab, which is approved for these allergic disease conditions, and inhibits free IgE binding to its receptors on mast cells and basophils, but it cannot remove pre-bound IgE on mast cells. Therefore, it cannot prevent degeneration from these cells until they disappear from the circulation. And clinical improvement in allergic asthma may take up to 12 to 16 weeks. So we try to come up with something that works faster by immunizing rabbits with the recombinant CF224 and pan the library against it and obtain a huge number of clones, 182 clones made of 15 clusters based on heavy chain CDR3 similarities. And there's a major cluster that most clones belong to, but there are many other minor clusters that are easily missed by biased PCR amplification and inefficient phage display. And the interesting thing is that the cluster 3 to cluster 15 clones are all from the bone marrow libraries. We often see this more clones, more diversity, and also better candidates from bone marrow libraries than from spleen libraries. That may be because there are more IgG producing cells and the plasma cells in bone marrow. That's why it is important to include bone marrow cells. So after evaluating these 23 clones, we identified these 3 from minor clusters as the best candidates. And these strongly inhibited the binding of IgE to Fc-epsilon receptor transgenic mouse mast cells better than the omalizumab, and also removed pre-bound IgE from these cells that correlated with the prevention of degeneration. They were also tested in a mouse model of passive cutaneous anaphylaxis, and these 2 clones strongly inhibited the allergic reactions in these mice, but omalizumab did not. The next example is antibodies to multi-transmembrane proteins such as GPCRs and ion channels by peptide immunization. We designed a peptide mixture for PAR1 and immunized rabbits with that and panned the library with the peptide and also PAR1 positive cells while eliminating binders to PAR1 negative cells in a negative selection. We obtained a huge panel of clones specific to PAR1 expressing cells from the peptide panned and cell panned libraries, and 3 of these guys showed strong inhibition of cleavage by thrombin, which is related to the activation of PAR1. And this method was successfully used for 3 types of GPCRs and 2 types of ion channels. The next example is an antibody to a nucleoside, an oxidative stress marker 8-OHDG. There's a commercial kit using a mouse antibody, but has been shown to cross-react with 8-OHG and 8-SHG and urea, which is found at high concentration in the patient urine samples. And so as a result, it produces values more than twice as high as those obtained by HPLC measurements with significant variability. So we try to make something that's more sensitive and specific than mouse antibody. We immunize rabbits with a KLH conjugate and either library and pan against the BSA conjugate, while eliminating all the cross-reactive clones in a complex section against these compounds found at high concentration, including these compounds in the patient samples. And we obtained really highly specific and sensitive antibodies compared to the mouse antibody. We filed a patent. And also, rabbit antibodies are really good at making PTM-specific antibodies. We have many phosphorylation-specific antibodies as antibody products on our website, and we usually immunize rabbits with a phospholipeptide and pan the library with the phospholipeptide while eliminating pan-peptide binders using non-phospholipeptide in a competitive selection. And we do get phosphor-specific binders by ELISA after peptide panning, but they do not always bind to the cells. But we can do cell panning using these stimulated cells, and we get many strong binders to the cells, and this big one became a JAK2 phosphorylation-specific antibody product. We also made a methylation-specific antibody against MAP3K2, and also sulfation-specific antibody against CCR5, and our collaborator published the Western blot data in these 2 journals. And sometimes our customers use our antibodies in immunohistochemistry. This is for BP1 phosphorylation-specific antibody, specifically stained the glomerulus. And also, this is an anti-myelin rabbit antibody, stained luminous surface of the blood vessels in an airway inflammation. The last example is antibodies used for TDM. This is used for evaluation of therapeutic efficacy, determination of optimal dosage, pharmacokinetic assessment. In this example, it's an anti-idiotype antibody that is specific to the therapeutic antibody antigen complex. So this is so-called type 3 idiotype antibody. We immunized rabbits with the complex and then tested the serum against free antibody and antigen, we saw stronger signal against free antibody. So it is not easy to obtain complex specific antibody just by screening. But with phage display, you can do negative selection with a free antibody and positive selection with a complex, and obtain these complex specific binders. And the purified antibody also showed really strong and specific binding to the complex, but no binding to free antibody or antigen or human IgG. And it became the product of a local diagnostic company. And the last example is also antibody for TDM. We have multiple projects for this company, and this is against antibiotic. And after the immunization with this target antibiotic, we saw strong cross-reactivity against these antibiotics that belong to the same class. But we did a complex selection using these antibiotics, and we obtained these two clones very specific to the target antibiotic, no cross-reactivity to these antibiotics that show really strong cross-reactivity in the serum analysis. And the binding was really strong and sensitive compared to the mouse control antibody that we obtained from the client. We also see higher affinity and better sensitivity from rabbit antibodies than from mouse antibodies. Okay, so we do the humanization ourselves for the therapeutic development. So we graft CDRs into human germline frameworks, and the CDRs are defined based on an antibody database, which is different from IMGT, CHOTHI, or CABOT. And we consider putative hypervariable region 4 in framework 3, and then sometimes do in silico immunogenicity analysis and use sequence liability prediction tools, and typically make 1 to 4 clones and express them as IgG, and evaluate them in ELISA flow cytometry, affinity measurement, and functional studies. And also do developability assessment by AC-SINS, BPP ELISA, melting temperature measurement, and size exclusion, HPLC, and others. So far, I have humanized 11 rabbit antibodies successfully and 4 from other species, and I show you some examples. So these are those anti-IgG clones that show really good binding and also removed efficiently IgG from the mast cells. And this clone had really high affinity as IgG and Fab compared to omalizumab. And this is another target for allergic diseases called histamine releasing factor HRF. Binding of IgG to HF dimer can lead to cross-linking of IgG molecules and mast cells and basophils that could potentially trigger allergic reactions. And so we obtained these 3 clones that strongly inhibit the binding of HIF to IgE. And this clone was the best one and humanized. All the humanized clones showed really good binding compared to the parent rabbit IgG. And this one showed pH sensitivity. And also these 2 clones prevented the body temperature in a food allergy model. And the last example is anti-COVID-19 neutralizing antibodies. At the onset of the pandemic, we immunized rabbits with the Wuhan Human strain receptor binding domain protein, and they got these 4 neutralizing clones. And they were humanized and sent to COVID consortium at the Hoi Institute for Immunology by Eric Safaya's group. And they also great binding to original strain and also D61-4G variant, and then also neutralize the wild type strain. And particularly this one showed good neutralization of beta and delta. We compared this clone to the parent rabbit IgG in a surrogate virus neutralization test using the recombinant spike primers and ACE2 protein. And this one showed really broad neutralization of Omicron variants. And then Kevin further engineered this clone using stage enhanced maturation STEM technology to obtain clones that broadly neutralize Omicron variants. And with this, I'm going to switch to Kevin for his presentation.
Okay, thank you. So I want to thank everyone for sticking around so far. And as a reward for your attention, I'll play a cool robot video. So this is just a minute long, but this shows some custom robots that we built. So back in 2019, we were looking to expand our capabilities, and we paired up with a local robotics company and custom designed these from the ground up. So they're really unique, in the field. And what you'll be able to see is there's basically a central robotic arm that accesses these. And so all the labware is in 96 well plate footprint, and this arm can add the plates to every single instrument on the robot. And then we also have a second arm you saw right there that will load the deep well plates into the centrifuge to spin down the cells or the phage. So these really increase our throughput and our reproducibility. But one of the biggest things is that they also reduce our lead time. So we have two different systems, and after the video, I'll show you the kind of footprint of the two different systems. And our panning system is one of the ones I'm most proud of. I don't know that anything exists like that out there. So, all right, let me go on to this slide. There we go. And so here's our phage panning platform. So, our panning used to be in flasks, and it'd take a PhD level scientist 6 hours a day. But now it's in 96 well plates, and it's fully automated. And so one of the biggest benefits, like Toshi mentioned, we can do 8 or 16 separate libraries and do that many different discovery campaigns if we want to. But what we often do is a single library with many different selection conditions. So instead of taking like a week to do phage panning and a week to do screen and seeing what you get out, you can do many different conditions, varying the antigen concentration, different antigens, different washing stringency or cross-reactivity clones, and really quickly find out the best clones, and you get a lot of information out of those campaigns also. So not only do we have phage panning, but we also have an automated Fab screening. It's got a lot of similar instruments, also a colony picker, and this can also automate DNA purification and some antibody purification. So one of the other big things that Abwiz offers is our antibody optimization platform. So we call this STEM because it's a strong core technology, but it's also very flexible. And we think this combines the best of both worlds in that we can use smart informatics-informed library design, but pair that with functional selection of the entire library rather than just relying on screening. So I'll go into how the libraries are designed a bit later, but the idea is that we're maximizing the diversity, and because we're using phage, we can access high diversity up to almost 10 to the 11 without compromising the developability. So the other benefit that we're using phage is that phage are super stable up to 80 degrees. So we can do thermoresistance selection so that we get clones that have are very stable, even improved compared to the parent, and better developability profiles. We also have polyreactivity filters to remove problematic clones. So we've used this successfully for many different applications, not only increasing potency, but also increasing affinity, do specificity engineering, as either selecting for or against cross-reactive binding to related proteins, or for proteins of different species, like both human and cyno reactivity. We've done kinetic selections, targeting specifically the on or the off rate, or to select for avid binders. We can do pH-dependent selections where we select, which is useful for either getting clones that target the tumor microenvironment or that are useful for antibody recycling. And we can also embark on campaigns to just improve expression or developability of a lead candidate. So our first campaign was back in 2016 at Abwiz. So Toshi and I had experience prior to that. And we most commonly have done this for human therapeutic antibodies that end up going on to clinical use. We've also done this with mouse, rabbit, even feline antibodies. And also it's a generalizable platform. So we can use this for recombinant proteins. And we've done this for a non-antibody as well to select for improved binding to its protein partner. So I'm going to go a little in depth about how the platform works, and then I'll show a more detailed case study. So in stage A1, we build 6 separate libraries targeting each CDR individually, and we can build these really large. So the purpose of stage A1 is to focus the library into functional CDR pools. So we apply those filters. Stability and polyreactivity is included throughout the entire campaign. And then we also apply functional filter. But in stage A1, we usually use a more mild functional filter, and because the purpose is to capture as much diversity as possible, and we want to get all the binders that are present. So usually after stage A1, we just do more limited screening just to confirm that we still have full-length clones in the library and we have good diversity, because where the real power of this technology comes is the next stage, where we combine those pre-selected CDR pools. So we'll amplify them from the selected phage and pair them by overlap PCR. So for instance, we can have the combined heavy chain 1, 2, and 3 CDR pools paired with the wild type light chain, or vice versa. And at stage A2 is where we apply the really strict functional filters and do exhaustive screening to find lead candidates. So we used to call this stage 1 and 2, but we paired them to stage A A1 and A2 to really drive home that it's this multi-step approach, kind of mimicking what happens in vivo with somatic hypermutation. When we want the ultimate, improvement in function, we'll even move on to stage B, where we'll take those combined heavy and light chain libraries that were selected from stage A2, pair them together in a single library for even further and more stringent selection. So our goal when we're designing the library is to maximize diversity without compromising developability. So because we're using phage, we can get up to 10 to the 11 almost in size, which is higher compared to other display technologies. And when we're designing the library, we'll take all the information the client has, but sometimes we just have the antibody sequence. And so we'll compare it to the closest human germline and then look at other characterized antibodies that share that same germline, and we'll look at what the amino acid distribution is at each position in the CDRs. And so we want to find positions that show high variability, those probably tolerate mutation, and ones that are conserved because this antibody database is from antibodies that have many different antigen binders. If we see conservation, that's probably important for stability. And so we don't want to change that. So we're not just doing random NNK mutagenesis throughout the CDR. Sometimes we do use NNK, but in other times we'll use other degenerate codons, or we'll even multiplex oligos to just mutate a certain position to defined amino acids. And we'll mix them in proportion to the observed diversity that we see during our bioinformatics analysis. So the idea is it's reflecting the observed natural human diversity. And not only are we getting better functional clones, but we're also further humanizing the CDRs. So the way that we build these is using synthetic oligos. That mutagenic oligo is often in a pool that are mixed at defined proportions, and we'll amplify that based on the wild type gene and use overlap PCR to create these separate libraries. So I'm going to go in depth into how we applied this to create broadly neutralizing COVID-19 antibodies. I think this it gives you a good example and shows the power of this technology. So Toshi talked about our lead humanized therapeutic candidate that came out of our rabbit campaign. And we built separate CDR libraries for this and did an initial selection on the wild type spike trimer. So like I said, this is more mild selection. And essentially what we're doing is just filtering out problematic CDRs and just retaining as high of diversity as we can of binders. So when we go ahead and from that create a combined light or heavy chain library, these libraries are essentially enriched for spike protein binders. And here, what we did was evolve our antibody as the virus itself evolved. So these selections were done on the variants as they emerged. And when we went ahead and created a combined light and heavy chain library, now this library is enriched for variants that are cross-reactive across multiple different spike trimers. And then this was selected using very stringent selection on the latest variants of the time to come up with the optimized therapeutic candidates. And so this is what we have published. And in the handout section, there's a link to the paper so you can see it in depth. And this is actually, 4 to 5 year campaign we've embarked on, where we're co-evolving a single humanized candidate and against the spike trimer. So anytime we saw loss of neutralization, we go back to our libraries. In this case, we actually took our lead candidates, created brand new CDR libraries from these, and then did selection on the later strains as they even emerged. So rather than going back and finding a brand new lead candidate that neutralized these later strains, we're just continuing to engineer. So I think it just shows the power of the platform. And so you can see these are IC50 neutralization values for a surrogate virus neutralization test. So we're looking at blocking of the spike protein trimer to ACE2. And, our lead rabbit candidate and humanized clone work pretty good against the Wuhan variant and then, early ones, but lost activity against Omicron. And we're able to recover the neutralization potency. But then anytime we come up with a new variant that evades, we're able to select for a new clone all the way down to our latest one, which was isolated in November and neutralizes the currently circulating dominant strain LP81 under conditions where you don't see neutralization by the parent humanized mAb or even, a clone that we isolated 2 years ago. So just one of the interesting things, and this shows, our capability. We have an octet, so we do BLI in house, and we looked at epitope binning, and all the way at least into the 2022 series that they retain binding to the Wuhan spike trimer, and by epitope binning, it didn't look like the epitope really changed, which was kind of interesting. It looks like it might have for some of the latest clones because we lose potency against those early strains. But then this has all been published in collaboration with La Jolla Institute of Technology. And there's a new paper they're going to come up with where they've done cryo-EM studies of this series of our engineered therapeutic antibodies co-crystallized with the spike protein variants. So you can structurally actually see both the virus and then the antibody co-evolve in real time. So I think that's going to be really cool. And they've come up with some interesting insights in that. So back to our platform, one of the things we really like to stress and to set expectations is that it's the multiple stages where we see the best improvement. And so this is an example where we have the wild type clone and then the top clones from L3, H1, H2 libraries that don't show much improvement compared to wild type. Here, the wild type was pretty weak, so we're only detecting binding in a bivalent pseudo IgG format. But when we go ahead and create our combined library and do much more stringent selection, we see significant improvement in binding when testing in a monovalent format under conditions where the wild type and stage A1 don't bind at all. And we've seen this over and over again. This is a campaign we just wrapped up a couple weeks ago where we saw the exact same thing. So there's a lot on this slide, but I think it's gives some nice takeaways, and I'll walk you through. So this was a humanized bispecific. So the light chain was fixed. We just did heavy chain engineering, and we came up with a bunch of clones that showed really significant improvement in binding by flow. So this looks at the library diversity that we designed. So if you're used to antibody sequences, this is going to look weird. It's because they're scrambled for IP issues, but the amino acid frequencies are preserved. So then you can look at your top 60 functional clones from the late stage. And what you'll see here is, first of all, there's a lot of diversity. So sometimes people like that because for IP issues. So you get a lot of functional diversity paired with sequence diversity that you can use when you're going after your IP. You could also pull a lot of information out of here. So you can get an idea about which residues were mutated but came back as wild type. Those are probably important for structure. Are there any residues that tolerated or preferred mutation to germline? Again, those might be stabilizing CDRs, and the benefit is they might be de-immunizing your clone. And then are there positions that were highly mutated but came back as a single dominant amino acid? So those are probably driving most of the improvement in binding that you're seeing. And we've even had clients take this information and then say, hey, we want, a brand new library across all the CDRs with just these dominant amino acids or reversions to germline to get the minimal mutations compared to wild type. So I'll go in a couple case studies that we've had success with. So like I said, most often we're doing affinity maturation, and this is just two examples. This is client data. This is actually our first campaign we were successful with to get a sub-picomolar binder. They ended up patenting the top clone from that. And then also we did a feline affinity campaign that resulted in a patent. We increased binding to single-digit picomolar over 1,000-fold. And the benefit or the interesting thing here was, I hadn't seen a report of a feline affinity maturation campaign in the literature or anything. So that was a fun project. So not only binding, but a lot of other functions we can go after as well. So this was an in-house one where we took infliximab and adalimumab, and we're engineering pH sensitivity to create a recycling antibody. So recycling antibodies will release their antigens quickly at pH 6 in the endosome, and that results in more efficient antigen recycling and higher potency for the antibodies. So you can see the therapeutic antibodies show almost no difference in pH 7 versus pH 6 dissociation, but our clones show really quickly dissociate from the antigen at pH 6, but still have really potent binding at pH 7, which is exactly what we were looking for. So we can also do this to tune kinetics. So in this one, this was an antibody that came from a humanized mouse, but the agonistic activity was too high. So in stage A1, we just did regular selection, an hour binding, normal number of washes. But then stage A2, we really varied the incubation times and washes, selecting for slow or fast on rate and fast or slow off rate. And we saw a real range in affinity from what we got out, where in the weakest affinity selection, binders, not very many binders when testing a monovalent ELISA, or the highest affinity selection, you see a lot of really strong binders. So it's you can get a real range of functional clones for there. And then we've also applied this to just improve expression. So this was a client that was happy with the activity, but their clone expressed really poorly. And so what we did was panning using cells. So that's another benefit. We can do either a discovery or an engineering campaign using just cells for our phage panning and for our screening. So here we really focused on our thermoresistance and polyreactivity filters, and the best engineered clones show significant improvements in the thermoresistance. This is a flow assay. Decreased polyreactivity, much higher expression. And then also we have an in-house sequence liability program, and we were able to remove that flags, potentially problematic liabilities, and we were able to remove those. And I think they ended up patenting the top clones from this as well. So then I want to go into a little bit about developability. So this has been a big focus for us because a lot of our engineering campaigns, both for clients and in-house, are looking to go towards therapeutics. So one of the nice things is that we're working on and have developed where we can screen for polyreactivity using the bacterial Fab supernatant. So it's nice because you can do that in high throughput in a 96 well plate. And we have orthogonal assays by both flow cytometry and ELISA to look at for polyreactive clones. And so you can kind of see the population. So this is looking at a population of clones from the same library that were selected for function, but then with or without polyspecificity subtraction. So you tend to see, lower polyspecificity from the clones that were selected without the subtraction. And when you look at these two different assays, they tend to pair pretty well. Some of the worst clones show really high polyreactivity in both flow and ELISA assays. And again, the best population of clones were actually selected by adding those filters during panning. And then this was for a Fab therapeutic. So we chose some of the best and some of the worst candidates from these selections, expressed and purified them from mammalian culture, and then retested them by flow and ELISA. And so when you compare the purified Fab and the Fab supernatant, you say they tend to correlate really well. So once you get down into the best clones, it's hard to rank, but you can at least bin some of the worst candidates versus some of the best ones. And we got clones that showed improvements over the parent Fab. So then we also have developability capabilities in-house where we can assess polyreactivity by BPP ELISA, high molecular weight species by HPLC. We do AC-SENS to look at aggregation. Again, I mentioned our sequence liability program and other thermoresistance or freeze-thaw studies. And then just a last example where we took this entirely in-house. So this was for infliximab, which has 74%. It's a chimeric antibody, so 74% of the framework residues are non-human, or are human. 26% are not human. So we went ahead and humanized that, and then applied that STEM technology. It ended up with engineered clones that showed significant increases in activity in a cell-based assay. And then also, when you look at both aggregation and polyspecificity, we saw we were able to engineer clones with really good improvements and that are looking better than the parent, both by function and developability. So that shows our capabilities. I want to thank everyone for attending, and that leaves us some time for some Q&A.
Hi, thanks again both for your presentation. Really, really informative, super exciting, and we are going to jump into our Q&A if you're ready. Okay, so the first question here is, do you lose the antibody affinity using phage display by selecting random matched H/L pairs compared to the native paired clones?
Well, we do not. Actually, it really represents the affinity or specificity what we see in the serum, and after the purification with the antibody. Yeah.
Okay.
So they're
Okay.
all really, as we shown in the presentation, they have really high affinity and specificity.
Okay, perfect. The next question here is, do you have data to show the antibodies from plasma cells are actually have higher affinities than from the memory cells?
Okay, because they go through more final rounds of affinity maturation, it naturally acquires higher affinity than the ones in the memory B cells. So yeah, that's kind of I think it's common knowledge, and those antibodies we put from the libraries compared with the bone marrow spleen, we also see many times we see better candidates from bone marrow libraries, and they contain more plasma cells also. And actually, one time we compared the bone marrow libraries and spleen and versus peripheral blood B cells, and even got associated lymphoid tissue gout. And we didn't get good clones from there, from the PBMC or gout. So that's why we stick with bone marrow and then spleen.
Okay, thank you. The next question here is for Kevin. Do you find that the flow MFI signal correlated with the antibody BLI binding affinities? Do you find that those two are correlated?
Oh, no, not always. So yeah, I think the sometimes the polyreactivity doesn't always correlate with the activity, but usually we get such high diversity that we have clones that have the high function we want and then also the low polyreactivity.
Okay, perfect. We've got another question here. What is the estimated time of development for monoclonal antibody using your technology for IVD or therapeutic intended uses?
But to get the initial candidates as become purified, become the IgG in a small quantity, it takes about 4 months. From there, we can do all the studies like functional studies or others, and we can narrow down to a fewer number of clones for humanization if it's a rabbit antibody. So for IVD, I think you don't have to do that. So it's faster. But for the humanization work, probably takes about a month or so. And again, we can do all kinds of studies, including developability assessment.
Okay,
So it
perfect.
doesn't take too long.
Mm-hmm. Awesome. Thank you so much for submitting all these questions. This is wonderful. We're going to keep going. Someone has asked, do you have access to IHC facilities?
Well, we don't ourselves. So it's usually our customers, use our antibodies and verify that.
Okay. Okay, perfect. Someone else has asked, do you have a transcription factor as a target? Is there any disadvantage of getting that?
Or a transcription factor as a target. We have never worked on that as a target before, but we can certainly work on any types of antigens as long as we can obtain immune response. Yeah.
Yeah, I don't know if this is relevant, but our phospho-specific antibodies, we do panning with the permeabilized cells against the native phospho target. So if it was a transcription factor, potentially you could use a similar approach for the selection and for the screening against the native, in addition to a recombinant peptide.
Okay. Okay, perfect. Awesome. We've got another question here. Are there binding or expression issues with phage display derived antibodies?
We never seen any, bad clones, really bad clones. Sometimes a few clones may not express, but generally they all tend to express very well as FAB or IgG. Like we can obtain actually better expression than mouse antibodies as we come to IgG.
Yeah, and then from the engineering side, so it's really the thermoresistance filter is a big one. I think the polyreactivity is very nice also, but the thermoresistance helps with any clones because we had a campaign where we did not include that and then had some issues with expression later on and went back and selected the same libraries with the thermoresistance filters, and that helps with the stability and expression issues that sometimes people tend to associate with phage display. But with the proper selection, then I don't think it's a problem.
Okay, perfect. The next question here is, does the extra disulfide in rabbit antibodies pose a problem when humanizing them?
No, not at all. They're contributing to the stability of the light chain, and it could be a substitute with alanine or serine. There's a publication that didn't lose the binding, but it just lost the stability in the rabbit antibody format. But
Okay.
when it's humanized, there's no issues about the stability or binding and affinity. So those are the examples that I show you in the presentation. They all attain good binding.
Okay, thank you. We've got a few more questions here, so I'm going to keep going. Is it necessary to go through all 3 stages of affinity maturation?
Yeah, so we have had clients where from the separate CDR libraries, they're satisfied with the affinity improvement and we'll just stop there. So at each step, we have a pause point and discuss all the results. And then we've also had ones where, we'll do the 6 separate CDR libraries. We'll see diversity in maybe 3 of them. And then, so we'll just proceed to a single combined library after that. So it's completely tailorable to each project or what each client wants. So,
Okay, perfect. Customizable. That's awesome.
yeah.
We've got a ton more questions, so I'm going to keep going. Have you ever developed a monoclonal antibody to detect a mutation?
Yeah. Yes.
Yeah,
Okay.
we have an active
Yeah.
campaign right now, and
Yeah.
I mean, like the post-translational modifications also show even if you have a very small difference between your target versus another one, we often have had success, and it's those rabbit antibodies are really powerful.
Yeah, we also have an antibody product detecting nuclear capsid protein of SARS-CoV-2 versus SARS-CoV-1. We used a peptide containing a few amino acid difference between the SARS-CoV-1 and 2, and the antibody specifically, detected SARS-CoV-2 only, and we did 2 regions, and we have a pair of antibody that can be used as a sandwich ELISA that does not detect SARS-CoV-1 nucleocapsid protein. So it's like a point mutation, and yeah.
And just briefly thinking of our current campaign, so we have one target with 2 different single point mutations we're going after. Serum screening is really important, and we also when we're going after something that specific, we'll immunize multiple rabbits and then screen all of them because sometimes you'll see one that has the best response and the most specificity. So as long as you have a nice serum screening assay ahead of time, that gives us a lot of confidence in proceeding forward with a campaign.
Yeah, if it is the target is or the application is Western blot, then definitely we need to perform Western blot to make sure that it binds to the target first of all, and then we see difference between the different point mutations.
Absolutely. Awesome.
Yeah.
Okay. Another question here, also kind of about mutations. Do you have a program to develop mutated CRAS antibody for therapeutic purposes?
Mutated cross.
The KRAS protein.
Okay. So yeah, if we can have that mutated one versus the wild type, we can definitely develop the antibody specific to that mutant.
Okay. Yeah. Good to know.
Yeah, like Kevin said, we just have to do the serum analysis properly to make sure that the rabbit produced something that could detect specifically the mutant versus the wild type. Yeah.
Okay, perfect. Another question here. How long does affinity maturation take?
So usually about we can go like 2 months for the stage A1 and stage A2, and then another 6 weeks for stage B. It's usually a campaign. It depends. depends on the project, but.
Okay, so just like a general range there. That's good to know. All right. Because of the time, I think this is going to be our last question here. But please continue submitting your questions. I will pass them along to the team at Abwiz, and they can answer your question via email later. So for GPCR target, what's your opinion for using peptide compared to VLP, cell, or other formats of antigens for immunization?
Well, as long as we can elicit good immune response against the target on the cells, I don't mind using any technology. So I've done VLP in the past, but we didn't see good immune response against the when we tested serum again in a flow cytometry. So we didn't use that. But with this peptide mixture approach, we got some really nice signals from the serum in a flow cytometry analysis, and we got this really good binders that reflects the binding we saw in the serum. So yeah, that's what worked for us. But of course, the cell immunization or VLPs or even DNA or RNA immunization, if those approaches work, then we definitely proceed with that live reconstruction from the samples.
Okay, fantastic. I just want to thank you both so much for your time today, for your presentations, and also your insights in the Q&A. It was a pleasure to have you with us, and I hope that you had fun.
Thank you.
In closing, we hope that you enjoyed this Scientist.com webinar produced in partnership with Inside Scientific and sponsored by Abwiz Bio, and I hope that you enjoy the rest of your day.